So let's begin by breaking down the word phytoplankton. So phyto means plant, which means that it produces its own food. And plankton means wanderer or drifter. And so in the context of the ocean, it means that it flows with the current. So phytoplankton put together, these are teeny tiny organisms that flow in the current of the water, and most of them produce their own food, which is pretty cool. But we'll find out later that not all of them do. They are members of the protist kingdom, which means that they're not plants or animals or fungi, but they belong in the same kingdom as other protozoa and cymodes, molds, and algae. And so they're eukaryotes, and that means that it has a distinctive membrane-bound organelle. And then something else that's cool about phytoplankton is that they produce most of the oxygen that we breathe, upwards to about 70%, which is really interesting to learn because most people think that all of the oxygen that we get are from trees. And while a lot of it is, most of it actually comes from the phytoplankton in the ocean, which is really fascinating. There are two different basic types of phytoplankton. There are dinoflagellates and diatoms. Dinoflagellates are distinct from diatoms because they have a transverse around their middle of their bodies, and that helps draw in food. Because like I mentioned before, some can produce their own food, but some will also collect from external sources, and some can do. And so there are heterotrophs and autotrophs, and then there are both. And so what is the flagellum? It's a protrusion from their body that kind of spins around, and for some organisms, it helps propel them forward and keep them backwards. And in the case of these phytoplankton, since they don't really have direction, they're wanderers and drifters, they use it to draw food in towards them if they're not producing their own. And phytoplankton are at the very, very bottom of the oceanic food chain, and we'll talk about that in a future slide. But so those are some of the basic fundamentals of what phytoplankton are. And as I mentioned, phyto means plant, and they have some cool things in common with plants. And so what are those things? They are able to photosynthesize. As I mentioned, they can produce their own food. So they use that chemical energy that they receive from the sun, and they're able to produce sugar from that chemical energy, much like a plant does. And as they produce their own food, they make a good resource for other organisms to use them as a food source. So they will be consumed by zooplankton. As you see here, they are called the consumers. And then those zooplankton will be consumed by small fish, and then the small fish will be consumed by larger fish, and so on and so forth up the food chain. Kind of similar to plants. There are many animals that will eat those plants, and then some of those small animals might be eaten by bigger animals, and so on. So those are some of the similarities. And so most of the phytoplankton that we see here are good guys. They're fine to be around, but there are a few species that are harmful that we don't really want in our water here. And here in Maine, there are 4 different types of phytoplankton that we consider the bad guys that are going to cause some kind of harm. And so what these 4 do is they can cause toxic shellfish poisoning in humans and other mammals. And so when these blooms, it's sometimes called red tide, but it's different than what you might see called red tide in Florida. And so in Maine, red tide corresponds to toxic shellfish poisoning, but in other areas, red tides may simply refer to a bloom in marine algae that turns the water red-brown color due to the concentration of photosynthesizing pigments. So depending on where you are geographically, red tide is going to have 2 different meanings. So it's always important to know where you are and the definitions of the same word in those 2 places. And so these are the 4 different types of bad guys that we look for here in Maine. And they all cause different kinds of ailments to people who end up consuming them. And so let me begin by, how do we get these phytoplankton in our bodies? Well, as they're at the bottom of the food chain, they will be consumed by different kinds of mussels and oysters and other shellfish that people will then harvest. And while the shellfish don't get sick from these toxins, the toxins do get embedded into their flesh, which we then eat. And then when we eat it, we ingest the toxins, and it does have a harmful effect for us, but not the shellfish. So the first one that we see is dinophysis, and this causes diuretic shellfish poisoning, which means you're going to get a lot of diarrhea and you're going to have an upset stomach for a while. The second type that we look for is Alexandrium, which causes a paralytic shellfish poisoning. So that one's a little bit scarier. And so it causes a kind of paralysis where you can't really move your body. So it's a loss of voluntary muscle function or sensation, which is caused by lesion or disorder of the muscles or the nerves that supply them. The third type is Protocentrum lima, which, like dinophysis, also causes the diuretic shellfish poisoning, so more upset stomach. And then the fourth kind is Pseudonychia, which causes an amnesic shellfish poisoning. So this is going to give people short-term memory loss, or in really bad situations with a high dosage, brain damage. So it's really important that we monitor for these four and we make sure that they're not in high abundance, and that if they are, we're able to send out warnings and let people know, don't harvest the shellfish right now because it can cause a lot of really bad health effects for the consumers, for the people. And so I shed a little bit of light on this already, but in more detail, how do you get toxic shellfish poisoning? Well, as I mentioned, the clams or the oysters or other bivalves gather the food by taking in water through their siphons, and then they filter out the nutrient matter, including the phytoplankton. Fun fact, a large oyster can filter up to 7 gallons of water a day. That's a lot for an oyster. And so phytoplankton contain biotoxins that's incorporated into the flesh of the bivalves and then ingested by us. Unfortunately, these toxins can't be destroyed by cooking, so even if you cook your clams or your oysters, the toxins are going to remain there. But something that's good to know is that you can't get sick just by swimming in the water. You have to actually consume the toxins. So it's not quite so dangerous to swim in them, but eating them is going to be bad. So what does it look like when we monitor for phytoplankton? There's a two-part component, and the beginning is in the field. And so we go out and we collect, first we get our water sample. And so how do we do that? Well, we have this 5-gallon bucket that we fill up to a specified mark, and then we pour it through this mechanism right here in this first picture, which is really just a PVC pipe with a really, really fine mesh in the middle. And so when we pour that bucket of water into this pipe, into this filtration system, the fine mesh will catch any phytoplankton that we have in our water sample, but the water will still be able to drain through. So we do that twice and we get 2 buckets worth of water. And then once all the phytoplankton and usually some other organic materials like mud and sediment will be trapped on top of that mesh filter. So then we take the pipe and we turn it upside down above a funnel. And then at the bottom of the funnel, we have a collection tube. So we're able to collect this phytoplankton. And we do so by taking another spray bottle that we've also filled with the same ocean water, and we spray it on the underside of that mesh, which is able to push the phytoplankton that's trapped on the underside down through the funnel and into our collection tube. And we fill that to 15 milliliters, and then that becomes our sample that we use in the lab to look at under the microscope. What else do? We collect some other water samples on the way so we can look at nutrients and what other chemicals or compounds might be in the water that could also affect the kind of phytoplankton that we see or other health concerns that we might be interested in. And then we also record observational data. So we look at the weather, the temperature, the wind direction, if we see a lot of pollen on the water, if maybe there's a lot of foam collecting on the shoreline, are there boats, are there fishermen out, are there people swimming? A lot of different things that could be really important to note when looking at the big picture once we're in the lab and analyzing all of this data. We also determine the transparency of the water. to see if there's a lot of turbidity or not. So if it's clear or if it's really murky and the sediment's been stirred up. And so there's a lot of different things that we do once we're in the field. But once we've collected all of that data, we go into the lab, so we're able to take a closer look at everything that we have. So one of the things that we do is we look at the determined dissolved oxygen levels, which is also called DO. And we can also do this in the field sometimes. We have a meter that can be put in the water and assess all of that data for us. But if we don't have that meter, we can do it by hand. And so we do that by adding different kinds of chemicals. I believe it's 8 drops of each at a time. And so we create the solution and then we titrate. So we take a different kind of chemical and we add it to the water until it becomes clear again, because adding the previous chemicals turn it kind of a yellow color or purple. And then we add another chemical to it until it's clear. And then we're able to take a measurement to see what the dissolved oxygen level is. But why is that important? Why do we care about the dissolved oxygen? Because all aquatic animals need dissolved oxygen to breathe. So low levels of oxygen, which is called hypoxia, or no oxygen levels at all, which is called anoxia, can occur when excess organic materials, such as large algae blooms, are decomposed by microorganisms. During this decomposition process, DO in the water is consumed. Low oxygen levels often occur in the bottom of the water column and affect organisms that live in sediments. In some water bodies, dissolved oxygen levels fluctuate periodically, seasonally, or even part of the natural daily ecology of the aquatic resource. So as the DO levels drop, some sensitive animals may move away, decline in health, or die. So DO is considered an important measure of water quality that's a direct indicator of an aquatic resource's ability to support aquatic life. So it's important that we take that measurement. Other things that we look at, we take that data sheet that we had in the field where we marked down all of our observational data, and we also record it on an electronic log on our computer, because it's always important to have your data in multiple places. So in case the paper copy were to get lost or get covered in water and you can't read it anymore, we have that other copy on the computer. And then finally, we also identify the different phytoplankton that we see under the microscope because they're so little. And so we're able to see if we have any of those 4 target species, and we keep a tally of every one that we see. Excuse me. And then we also record the most abundant phytoplankton that we find, whether it's a harmful one or one of the good guys. Because phytoplankton, much like other plants, will show up in abundance with the season. So just like some flowers will bloom in early spring and some will bloom in later spring, phytoplankton occur in the same way, where we'll see an influx of one in early spring and then different ones in maybe midsummer. And that can also indicate seasonality change and can help us see if there are other global impacts that are changing when they show up in our water. And maybe if the water temperature is colder or warmer than it was the year before. So it has a bigger impact to us than just human health, but also more environmental health as well, to give us a sense of what's going on. And so when we look at these phytoplankton under the microscope, there's different ways that we can analyze this data. And you can see here, there are different kinds of graphs, and these are different ways that we can help visualize what's going on in the water. Because it can be a little bit hard to understand sometimes if you're just looking at numbers on paper. But when you have visuals like this, where you have different kinds of colors and you can see one part of the pie chart is a lot bigger than the other parts of the pie chart, it's a lot easier to comprehend what is really going on in the water and who's in more abundance and who's lacking numbers. And then we can look back over previous years, because we save all of this data, copies and electronically, and we can see if it matches up to previous years or if we're seeing a huge change in one kind of species over another. And this again can help us look at year-long trends and what's normal and what's changing and why that might be happening. Do you have any questions? Is there anything you need me to repeat? Sorry if I went a little bit quickly. but I'd be happy to answer any questions now if you have them. Or comments or something you thought was really interesting, or maybe something you didn't know that you are excited to share with your friends and family. And if not, that's okay too.
Anyone? That was awesome presentation. I felt like I learned a lot.
Thank you.
Yeah. Oh, we have one question coming in the chat.
Okay.
Brianna said, I studied this in grade school and part of high school. Yeah, so you guys did outreach to schools. That's right.
Cool, yeah. So we also teach some schools. We'll have some elementary schools come in, sometimes middle schools. In the past, we've let them help us do phytoplankton monitoring. And so we'll take them out to the shore next to the lab and show them how the process is done, and we'll let them have the hands-on experience too. So that's awesome that you've learned about this already. That's super cool.
Mm-hmm. Well, let's see. I wrote down some questions.
Awesome.
Let's see. Oh, so you were talking about how some phytoplankton sort of arrives seasonally. But I kind of remember, I actually think I'm in one of those slides as a young child.
Oh, really?
Yeah. But I remember that we would often see dinophysis, but it's not toxic all of the time. So, yes. Right. So sometimes we would see one of the toxic phytoplankton, but they're actually only a problem when they're in bloom. I'm forgetting how you tell.
Yeah, that's a great question that I was debating if I was going to get to or not. But so two that we see often that aren't always dangerous, dinophysis and pseudonychia. And part of that is because there are subspecies of these. So there's dinophysis, but you can think of it as like dinophysis has some cousins and they look similar, but not all the cousins are bad guys. Some of them are fine. They just look like the bad guy. And so there are really fine distinctions between them. And so we have this kind of like a cheat sheet with all the different species, and it shows the subspecies of the toxic ones. And so we're able to use that to see if we're looking at a bad guy or a good guy that looks very similar. But they generally have the same shape. Sometimes one of the differences in dinophysis is this bottom part here that's pointed. One subspecies has a more rounded bottom and the others is slightly more pointed. So they're really, really fine distinctions that you have to look at really carefully. So sometimes we'll take a picture of our phone through the microscope and we'll send it off to someone who's a little bit more experienced in this ID if we're feeling uncertain. In like pseudonychia, sometimes we'll see really, really small chains of them. And what I mean by chains is you see all these independent segments and then they're linked up. So sometimes there's maybe only 3, and then sometimes there's 27. And so pseudonychia difference here is also really tricky, where the width is off by just a little bit. One is a little bit more narrow than the other. And so for that, we would really need a more advanced microscope that allows you to track the width of the different species, or sometimes you can pair up a microscope with a computer and you can actually draw a line segment over the width and the computer will tell you how many microns that is. So very fine distinction. So it can be really tricky.
Wow. Yeah. So the images we're seeing on this slide here, they're very magnified, right? About like 200 times. This is like under a microscope, just that making sure everyone understands how microscopic these organisms are.
Addie, I sent you on chat.
All right, let's check it out. So we have 2 questions. Darian's asking, is there any way to get involved with phytoplankton monitoring? And Paula's asking, can you get a rash from red tide? And I know that Paula is coming from living in Florida for most of her life, and Katrina lives in Florida currently, so they have a different sort of red tide down there, like you were mentioning.
Yeah, so up here in Maine, I haven't heard of anyone getting any rashes or external problems just by being surrounded by the phytoplankton. So these kind here, you really have to ingest to get sick, but swimming in it would be fine. In terms of getting involved, this summer would be a little bit tricky just because we're not really taking volunteers because of the COVID-19 situation, but we have been really excited to have volunteers in the past. And so what I would suggest, if you were still interested this summer and if it looks like things are you know, better in terms of COVID right now, then there's a good chance that you could definitely help out. I would suggest you could send me an email and I can pass it along. So my email would be awolf, like my last name, with an A in front of it, at mdibl.org. And I would be happy to pass on that information and see if we could find a place for you to help out, because we love citizen science and we love getting people involved. We think it's a really great way to, you know, build community, to share science and help educate people. So that's awesome.
Any other questions from anyone? Gabe, did you have any questions?
No.
Yeah, for sure. That was a lot of information to take in. But it's
if it would be helpful, I could send the PowerPoint to you too, and it has some notes at the bottom. If I went over anything too quickly and you want to really break it down, I'd be happy to send that along.
Yeah, I didn't feel like it was too quick, but definitely
Okay.
reviewing, it would be awesome for us to go over things. Yeah, especially.
Awesome.
Yeah.
Yeah, I'll do that right after this call.
Oh, I had one more question.
Yes.
How many sampling sites do you have? I was wondering if you see toxic phytoplankton maybe off of one beach, right? Does that necessarily mean it's at a beach on the other side of the island?
Right, good question. So we sample at 2 different places. So we'll sample off the dock at the biolab, and then we'll sample off the dock in Bar Harbor. So they're a few coves away, so not everything will flow through. And we don't always see, we don't always see the same concentration of phytoplankton. When it's seasonality, or when it's seasonal, sometimes we'll see there's an abundance of the same one in both places. But we will find that sometimes one localized area will have a different primary phytoplankton at one point in time than another area will. So it does vary by location. Even if they're not super far apart, we will see some differences.
Very good. Great. So
Awesome.
No other questions or things that stood out to you? Anyone's welcome to write in the chat or say something out loud. Darian?
I do have a question. Instead of writing in the chat, I'll ask. How long have you been doing this for?
Good question. So I started helping with the project in 2015. So I've been doing it for a while. In terms of the project as a whole, that's a really good question. I don't actually know how many years it's been going on. Certainly a lot longer than I've been participating. But yeah, so I've been helping out for about 5 years.
Wow, good for you.
Thank you. Yeah, it's been a lot of fun, and when we have volunteers, when the summer allows for it, I get to help in the training process and teaching people how to do it. So it's a lot of fun. Good question.
Nice. Well, great. If everybody is feeling good and there's no more questions, we'll definitely take some more class time and start to reflect on everything we learned. Amber, I don't want to take up too much of your time today. We really appreciate you coming in.
Of course, I was super happy to share some knowledge. So glad I could participate with you guys.
No, it's awesome. We've been looking forward to it all week.
Awesome. Awesome.
Yeah.
All right, cool. Well, if that's everything, I'll leave you guys to the rest of your class. If you think of any other questions, feel free to send me an email. I'd be happy to get back to you.
That sounds great. Thank you so much.
Awesome. All right. Bye, guys. You were a great class. Thanks for all your great questions and for listening.
Great. Thanks so much, Amber.